Hey everyone, welcome to this video on PCR. PCR stands for Polymerase Chain Reaction. It's used in molecular biology to take a single copy of DNA and make many copies of it. If you ever enter a molecular biology lab, scientists are literally always doing PCR. It's almost a miracle to find an open machine if you're trying to run your own reaction. But in this video, I'm going to talk about the reagents that go into making a PCR reaction successful, the theory behind it, and what you would actually have to do to set up a PCR machine if you are running it in a lab. So let's get started. Let's talk about reagents. What do we need to put into our reaction to actually make it work? Let's start with a list. So we have primers, polymerase, DNTPs, and of course, DNA. So let me talk about each of these. Primers. Let's say, for example, you have a double-stranded piece of DNA. Let's say this is the 5' end and 3' end of one of them, and the 3' end and 5' end of another. So, in the first step of PCR, which is a process called denaturation, what essentially happens is this DNA sequence is denatured or split up from a double-stranded piece of DNA to two single strands. So now that you have a single strand, it allows for the second step. And what the second step is known as is annealing. Annealing is essentially binding. And so let me just change the color. What happens in the annealing step is the primers, which are essentially DNA sequences themselves, are able to bind to certain regions of this single-stranded piece of DNA. So let's talk about what primers are. A primer's job is to point out what the target region is. And what I mean by target region is the part of the DNA sequence that scientists want to amplify. So scientists design these primers in a way that will get them that target sequence. And so since they know the exact sequence of the DNA that they want to amplify, they can design these primers. And what I mean by design is create this exact complementary strands to the DNA sequence that sit right outside the DNA sequence they wish to amplify. And so right here in the circled region, say if that's the DNA strand I want to amplify, then I want to make my primer, which is right here, sit right outside that region. Because what a primer does is it essentially acts as a flagger. And what I mean by that is it is responsible for telling polymerase where to come and start its DNA sequence. So let's pretend this is polymerase. And so what polymerase does is it comes and binds its huge machinery right on top of this primer region. And so what that allows it to do now is to say, okay, now I'm latched on, let's start adding nucleotides to this sequence in order to create another sequence that is complementary to my double strand. And there's a problem here. Like in each cell in your body, there are no free nucleotides floating around that polymerase can just grab and add to the sequence. So we're going to have to put them in. And that's where DNTPs come into the picture. And so what DNTPs are a mixture of DATP, DGTP, DCTP, and DTTP. If you've studied DNA to any depth, then you understand that there are 4 nitrogenous bases, and they're right here. A for adenine, G for guanine, C for cytosine, and T for thymine. And so what DNTP actually stands for is deoxynucleoside triphosphates, and they're in a form that this polymerase can add to the sequence right here as it continues down this path and this path over here. So essentially, what that process is called, that's the third and final step of the PCR reaction, it's called elongation. And so this polymerase is able to move in this direction, adding nucleotides to the DNA. And so essentially, out of one double-stranded piece of DNA, you're able to generate 2 double-stranded pieces through this process. And so as PCR keeps going, the DNA that is generated, for example, these pieces, are used as templates for subsequent replication. Alright, so I'm just going to erase some stuff to make some space. So let's talk about temperature, which is one of the most important variables in the process. The most common machine used to carry out the replication process is a thermocycler, or a PCR machine. So what a thermocycler does, essentially, is change the temperature between hot and cold over and over and over again to carry out these 3 steps in the process. So why does it do this? Well, because there's an optimal temperature for each step of these processes. So let me just go through the 3 steps once again. The first step, denaturation, is pretty self-explanatory. You need to denature or split the double-stranded DNA into single strands, like I did in this section right here. I took the double-stranded DNA and split it up into 2 single strands. After that, let me just change the color. After that, what I was able to do is add primers. I was able to bind the primers or anneal them right here and right here. Now, once the primers were added, the next step was the actual elongation process. And so the elongation is where the polymerase is able to add the nucleotides all the way through this reaction. And so as you can see, there are different reagents that are used in each of these processes. So let's talk about some optimal temperatures for each of the steps. Denaturation, I just need to make it really hot because DNA is a very stable molecule, but I need to split up that DNA. So I need to increase the temperature to a very high amount. So an usual temperature that's used for denaturation is about 94 to 96 degrees Celsius. Now, after I've split up the DNA, I need to anneal it. And so I need to bring that temperature down to where the primer is functioning optimally. And so each primer has a very specific melting temperature. And an annealing temperature is usually about 5 degrees below that melting temperature for that primer. So now, once the primer has binded, I'm able to elongate. And so for the elongation process, I need to have the temperature set to the optimal temperature for my polymerase enzyme. And usually what that is about 72 degrees Celsius. And so by varying my temperature over and over again, I'm able to create these multiple copies of DNA. So let's talk about cycles. And what I mean by a cycle is these three reactions. Each cycle consists of all three of these reactions. So when it comes to deciding how many cycles to run, it depends on the amount of DNA you want as a final product. The more the DNA, the more copies, the more cycles you have to run. And so the typical polymerase chain reaction has about 25 to 30 cycles. So let's talk about what this is useful for. Why do we want to do PCR? What's the point of all of this? Well, PCR has a variety of uses. It's involved in DNA cloning, analysis of certain genes, if you're trying to diagnose a hereditary disease, or even in forensic sciences. The reason for this is it's important to have a high concentration of DNA for a lot of molecular biology analysis. And PCR gives us the ability to make an infinite amount of DNA out of a single strand in theory. My molecular biology teacher always used to tell us that she would literally fail us if we asked for more DNA, because you should never ever run out of DNA with PCR as long as you have a single strand. It's almost like magic. Well, anyways, that's all I have for you guys on PCR. If you have any questions, please don't hesitate to leave them in the comment thread. Thank you so much for listening.